All right. Good morning, good afternoon, good evening, wherever you're joining us for this webinar on drugging the dead, selective targeting of pseudokinases. I'm Natarajan Kannan, professor at the University of Georgia, and also a member of the IDG, Illuminating the Druggable Genome Consortium. So we have an exciting line of speakers, really going to discuss some of the emerging themes and progress in targeting the druggable pseudokinome. But before we get started, I just wanted to go through a couple of sort of housekeeping items. So we have 4 talks, each for 20 minutes. We'll have 5 minutes for question and answers after each talk. And you as attendees will be muted, so you will be able to post your questions in the chat box. And me and my co-organizer, Stefan Knapp, will try and coordinate the questions from the chat box. And the panel members will be able to read the questions and answer them for you either in the chat box, or they will answer you during the panel discussion, which we've planned for this webinar at the end. So without further ado, I want to get this webinar started. My colleague and co-organizer Stefan Knapp is going to get this started by giving an overview of the field, where we're headed in terms of targeting the pseudokinases and some of the challenges in understanding the non-catalytic functions of kinases and pseudokinases. And Stefan Knapp needs no introduction to all of us who work on kinases. He's a professor at the University of Frankfurt and a member of the SGC. So Stefan, if you want to share your screen and just give us a quick introduction.
Thanks a lot, Kannan. It is a real pleasure to introduce this meeting. So I hope you can all see my screen. Is that correct?
Yes. Mm-hmm.
Okay, Kannan already mentioned we have a really nice lineup of 4 talks discussing pseudokinases, and I think this is an extremely interesting area in drug development. It's really emerging area. And as you know, there are now several initiatives that try to systematically provide tools to enable drug discovery. I mean, one, of course, is the Illuminating the Druggable Genome, looking at the dark kinases. We have the UV Open network in Europe that will work on covering about 1/3 of the druggable proteome by the next 5 years. And then you saw in the right corner, and this is the part of the seminar series, the Target 2035, an initiative that tries to cover the druggable proteome until 2035. So I think with pseudokinases, we are looking at a really exciting area. There's about 10% of all human kinases that are catalytically inactive or poorly active. And what is interesting is that many of them have a very strong genetic link to disease development. So obviously pseudokinases have, apart from non-catalytic functions like shown here, important allosteric regulatory roles. So in principle, they can be targeted allosterically, and since the very successful development of, I would probably say, the most advanced pseudokinase inhibitor targeting the pseudokinase domain of TYK2, BMS put that definitely on the map that we can effectively target pseudokinases with a very good clinical outcome so far. So I'm really excited about this program that ended now phase 3 with very good overall response in multiple immune-mediated diseases. So how do we target the dead? So of course, I just saw an example of an allosteric inhibitor. It seems that the ATP binding is required for many pseudokinases. So it's an important component to exert their allosteric and regulatory function. But of course, there are other ways. We're just releasing a chemical tool for this pseudokinase called CASC. So this is a very unusual one. that has context-dependent residual activity under conditions where we have low metal content in the cells, like very often in neurons, you have very low magnesium concentrations. And under these conditions, this pseudokinase is active. And you see here, it has a highly degenerated DFG motif, which is GFG. So it's missing the important D that coordinates the ATP. And this, and even the residue beside, is another small residue, another glycine, opens also a unique pocket. And I think that's one of the learnings, that we can use the non-conservation of usually highly conserved motifs to make very selective inhibitor. And this compound that is soon available on our webpage, but not yet published, is one example. We target this pocket by these decorations in the pending ring system that only bind to this, that allows binding of this inhibitor to this very unique pocket. Then, of course, recently also a new, I think, quite exciting technology emerged. These are PROTACs, so proteolysis targeting chimera, principle bidentate molecules that recruit targets to the ubiquitin system, causing selective degradation. And here you see one example of E3 E1 E2 complex recruiting a target. In that case, it's a small protein called BRD4 to the E3 ligase VHL via the divalent compound that binds on one hand to the VHL, on the other hand to this target protein. So this has been exemplified for pseudokinases, for instance, for HER3, but of course, if we don't manage to make allosteric inhibitors, very often a difficult development process. If we manage just to target the active site with maybe ATP competitive compounds, we can use such a system to also simply degrade the proteins. So I think several options to target them. Definitely very good rationale in terms of genetics and functional studies in diseases. And so I'm looking now forward to the talks that we have. And it's my really great pleasure to introduce our first speaker, which is Patrick. So Patrick Ayers is a professor of cell signaling and head of the Department of Biochemistry and System Biology at the University of Liverpool. And he has a long-standing interest in pseudokinases, in particular the triples, but he is quite broadly interested in this area. And I'm looking forward to his talk, and he will present data on the pseudokinase PSKH2 as an emerging target from the dark. Thanks a lot. Patrick, looking forward to your talk.
Okay. Thank you very much, Stefan, and welcome to everyone who's joining us from around the world. It's great to, although I can't see you, to know you're there. So the subject of my talk is this unusual pseudokinase PSKH2, which stands for protein serine kinase H2. And my acknowledgement slide really helps you understand this is a team effort. We're working as part of the Illuminating the Druggable Genome Consortium with funding from the NIH and from BBSRC. And my group based in Liverpool is led by Dominic Byrne and Kritika Ramakrishnan. We've been working with Professor Kannan and his graduate student, Annie Kwon, and currently we're doing a lot of interesting things with pseudokinases with Safal Shrestha. And of course, we're delighted to work across the Atlantic and of course within Europe as well. So it's a delight, I'm sure, to speak to many of you from continental Europe. So let's get on with it, shall we? So we think of the human kinome really as a development of the work of Manning and colleagues from 2001. You'll all be aware of the tree. Stefan's already shown you this. There are approximately 60 human pseudokinases encoded by the human genome. And pseudokinases are really a computational explanation for amino acid sequence evolution. Kinases, as we've known for many years, largely from the work of Hanks and Hunter, but of course from anyone who's ever worked on an active kinases, have a number of classical amino acid motifs that are involved in ATP binding, so the glycine-rich loop and the beta-3 lysine, which coordinate ATP, the DFG motif, which binds methyl ions, as we heard from Stefan, or not if it's absent, and the catalytic aspartic acid in this HRD motif. And together these form this wonderful enzyme superfamily that bind ATP and then transfer gamma phosphate to substrates. Now, pseudokinases are different, and we recognize this, and it was recognized by Manning early on that they contain changes to these canonical motifs, and that is predicted to prevent either ATP binding or transfer of gamma phosphate. I think we think about this much more nuanced manner now, because many pseudokinases have either low activity or they bind ATP. And some pseudokinases have actually atypical chemistry, which allows them to do unusual post-translational modifications. And I think the pseudokinase field is really informing the kinase field, because we should also remember that many kinases operate in catalytically inactive conformations, which still drive very important biology and are still druggable. Okay, let's try and move this on. So we were amazed to find in one of our earlier collaborations with Kanan and his group at UGA that pseudokinases are not just found in the well-studied high eukaryotes, like in mammals and the model systems, certainly the human and chimpanzee genomes, but that pseudokinases are found throughout all kingdoms of life. And the number is relatively stable in higher eukaryotes, where about 10 to 12% of all kinases, and these are shown in blue on this figure, in orange on this figure, are pseudokinases, and the rest are recognized as kinases with high catalytic potential. But this is also true through birds, arthropods, through non-vertebrates, and then into the other major kingdoms of plants, fungi, and protists. And indeed, many prokaryotic species also encode pseudokinases in their genomes. It's slightly more variable, but both bacteria and archaea are essentially full of pseudokinase-like proteins that have likely very interesting and atypical signaling functions, which are likely to be druggable, as Stefan has mentioned. So human pseudokinase domains are important drug targets. You've already heard a little bit about the JAK2 kinase domain targeting. HER3 is probably being targeted either knowingly or more likely unknowingly with approved drugs in oncology. It may be important in drug resistance. And we have a research program trying to understand how triple pseudokinases might be drugged with small molecules to help understand the pathways they control in cancer inflammation. You'll hear about MLKL from James in another forthcoming talk, but I just want to draw your attention to this pseudokinase GCN2, which has a kinase in the pseudokinase domain, and which is really important in stress responses. And we think that the pseudokinase domain of this kinase, which is always really studied in the IF2 response, is also going to be important biologically. But the focus of today's talk is PSKH2. I'm going to tell you straight up front, we don't really know what PSKH2 does in the organisms where it's found. But we've started a cell biology program to try and pick apart its binding partners and to try and get an idea about what it might be doing biologically. And of course, this is part of a bigger effort to try and understand how you might drug this and other pseudokinases. So nearly 10 years ago, James and I wrote this little mini review in Biochem Soc Trans, and we identified 16 human pseudokinases that lack this putative catalytic base, which I'm showing here with my pointer. It's also shown in red. This is found embedded in the catalytic domain and is a part of the catalytic loop. But as you can see, the absence of an aspartic acid at this position does not preclude conservation of other catalytic amino acids. So the beta-3 lysine is maintained in all these pseudokinases. There is a glycine-rich loop, which in many cases is functional and binds nucleotide. And the aspartic acid in the DFG motif is nearly always conserved. So these human pseudokinases are actually rather common, and extremely common are those that lack a catalytic base. So how are we going to study PSKH2 in context with the rest of the kinome? Well, it's certainly understudied. Until very recently, there were no publications that really looked at PSKH2 as a target. However, we are lucky that there are proteomics-based studies and transcription-based studies that have looked at PSKH2 and published some of their data, and I'll mention a little bit of that later. But PSKH2 is chronically understudied, as is its closest parallel, which is PSKH1, which was cloned in 2000, as I'll tell you in a minute. And protein serine kinase H1 and 2 are part of the CAM kinase family, and i.e. they reside in this arm of this branch of the kinome tree. And They are distinct from the AGC kinases, which we probably studied most of all, but they have lots of features that make them rather similar to classical protein kinases, which I will mention. So I'm just going to show one slide from Kanan's work, which I think is really intriguing, allows me to set the scene nicely, which is that PSKH2 conservation across species is unusual, even for a pseudokinase, because in higher mammals, and mostly in the primates, we note that the PSKH2 gene is normally present. There are a few species where it's not found, but that in all species where there is a PSKH2 homologue, it's a pseudokinase. So it has an HRN motif instead of an HRD motif. And when Safal and Kanan's lab has modeled PSKH2 based on the CAM kinases, all of the amino acids we would predict to be involved in nucleotide binding are present and stereochemically correct. The glutamic acid from the C helix, the beta-3 lysine, and this ASNA conformation, which should bind metals. What's really missing is the catalytic aspartic acid, where it's always an asparagine in higher eukaryotes of the primate lineage. Interesting though, in other mammals where there clearly is a PSKH2, and this is alongside a PSKH1, we don't have a pseudokinase population, and D is prevalent in the catalytic loop. So we would predict de facto that most mammals have a kinase PSKH2, whereas all primates, which of course includes humans, have a pseudokinase. So this is a really interesting example of something that's worth studying and has really been missed by the majority of people working in this field. And I should state that's because we don't largely have the reagents to study many of these kinases and atypical pseudokinases. So PSKH2 and PSKH1 are closely related. We know a bit about PSKH1. It looks a bit like this in simple cartoon format. It's got an N-terminus, which targets it to the Golgi apparatus. It's both myristylated and palmitoylated, and it has a C-terminal tail that binds to calcium calmodulin. And this is able to interact through the kinase domain to autophosphorylate it on the C-terminus. The PSKH1 is a Golgi localized kinase. Its biological function still remains rather obscure. It has about 83% similarity to the vertebrate PSKH2 of the kinase family. So these are the non-primate PSKH2s. And what's really missing in PSKH2 is this Golgi targeting motif. So in the vertebrates that have an HRD motif, obviously they look like they're catalytically competent. But in all the primates, PSKH2 has degenerated down to an asparagine, and this is an invariant asparagine alongside a couple of other amino acids, alongside this myristylation and palmitylation site. There is no Golgi targeting motif that we're aware of in PSKH2. So I'm going to spend 30 seconds on PSKH1. It's an interesting protein in that, again, it's a dark kinase. There's almost no studies in the last 20 years on PSKH1, apart from high throughput information from mass spectrometry. But as I've said, it localizes to the Golgi apparatus, and this work was shown by the Prids group in Oslo in the early 2000s. And that localization is, and it's shown here in this picture where they've expressed a flag type PSKH1 and co-localized it with beta cop in the Golgi. That localization is dependent on N-terminal palmitylation and myristylation. And those sites, as I'm going to tell you, are conserved in PSKH2. So how are we going to study PSKH2? Why is it important? Well, as part of the IDG, we really thought that an evolutionary analysis of PSKH2 could tell us something not only about PSKH2, but other pseudokinases and kinases. And I think that's turning out to be the case. In Liverpool, we focused on PSKH2 proteomics, an analysis of post-translation modifications and some of its cell biology. And really this work is the precursor for structural and chemical biology analysis of PSKH2. And really PSKH2, I think, provides a really great model for mapping really how pseudokinases have evolved or how they've disappeared from genomes and what biologically that means. So I'm going to start with the bad news. The bad news is we've spent many years trying to work with recombinant bacterial overexpressed PSKH2 to do some simple biochemical analysis. And we really have found that regardless of where we place tags or what we co-express PSKH2 with, we can never make folded non-aggregated PSKH2. So that's allowed us really to focus on expression in human cells. So we've taken the HEC293T model system to look at the expression of either the full length protein, and that's PSKH2 here with the pseudokinase domain, this N-terminal region, which we believe is myristylated and palmitoylated, and the C-terminal tail. And the very first experiment we did is to tag the C-terminus with an epitope, so it's MYC in this example, and to show we can express PSKH2 in the whole cell fraction. So all the studies I'm going to show you are whole cell extracts. We don't, unless right at the end I'll show you a little bit different, but these are whole cell extracts. And PSKH2 is expressed if we add MG132 to try and see if it's processed through the proteasome. We never see any effect at all, which we were kind of surprised at initially, but this is consistent across the board. Another pseudokinase we work on, Tribbles2, is massively stabilized by proteasome inhibitors like MG132, so there's nothing wrong with our drugs. But interestingly, we observed that if we delete this N-terminal region, we never really see any expression of PSKH2, except for tiny, tiny amounts. So PSKH2 expression in human cells seems to require the N-terminus. And a full deletion analysis across both the N-terminus, shown in this bottom slide, and the C-terminus really made clear that if the N-terminus wasn't present, we never see expression of PSKH2. So here's the two full-length proteins. We've changed the myristylation site here. It's still expressed. If we delete the C-terminal tail, which is amino acids 320 to 385, we call it delta C here, we get nice expression of PSKH2. But essentially, any other deletions that involve the N-terminus, we lose the protein. We cannot make stable protein. So we thought it was time to do an interactome analysis with human PSKH2 because we have a lot of the reagents. We collaborate with Claire Ayers' lab in Liverpool. And so we generated some new constructs for this where we could do the cleanest type of immunoprecipitations possible. These are strep-tagged PSKH2 at the N or C terminus with a strep-tagged green fluorescent protein control. And we've transfected these cells. We've lysed the cells in mild detergent, but 1% beta-maltoside. And we've then immunoprecipitated the proteins, cleaved with 3C protease, and then done a triple digest with proteases, trypsin, chymotrypsin, or elastase, either to do binding partner discovery through LCM-SMS approaches, and this gives us 123 confident PSKH2 partner proteins, or we've done phosphoprotein analysis, which gave us a couple of novel phosphorylation sites, which I don't have time to talk about today, but they are found either in the N-terminal region right next to the start of the kinase domain or within the kinase domain. We never get sequence coverage at the N-terminus, and we believe this is because of those myristylation modifications. In fact, we can't see them when we look for those as PTMs, as variable PTMs. But what I'm showing you now is to summarize a lot of work that we've done with proteomics and interactome analyses. We're actually going to combine our data with a data set that was published recently by Bojan and colleagues in last year. And they also did an interaction analysis of PSKH2 as part of a much broader study. And we all came up with the same major partner proteins, heat shock protein 90, CDC37, and FKBP5. And initially I found this rather a bit of a downer because I thought, well, most kinases are known to interact with the HSP90 system. But given that multiple groups have found this, alongside serine palmitoyl transferases and this protein UNC119A, which binds N-terminal meristylated proteins, we decided to go with this data. I'm also going to tell you one thing about mitochondrial membrane proteins, which is this protein cytochrome C oxidase, which is also found in this complex. So I'm just going to carry on. My computer's about to go to sleep, but I will just walk next door and plug it in. My apologies for walking you through my house. So to summarize, those are the main proteins we found in our analyses. We then went back and looked in published data sets, and these are shown here. These are the Bojan study, but also work from Hutton et al. And early work from Susan Lundquist to actually reveal two major protein interaction networks that PSKH2 appears to be involved with. The HSP90 CDC37 complex, and this mitochondrial complex, complex 1 of the NADH-UP-quinone system of mitochondria. So we thought this was worth focusing on. Just to remind those of you that have forgotten, HSP90 CDC37 is really a quality control system for protein kinases and many other enzymes, including ubiquitin E3 ligases and transcription factors. And what the CDC37 HSP90 complex does is to bind to a conformation of the kinase, or in this case, the pseudokinase, which allows it then to be either refolded or fully folded to attain a conformation that's most appropriate and that no longer interacts with this system. And that's done through ATPase cycles of HSP90. Now, when we went back and checked the early type Ale data, which was done in Lundquist's lab in 2012, and I'd never noticed this before, we were amazed to find that two of the best hits that were found as HSP90 binders were PSKH2 and PSKH1. So they have these HSP90 interaction scores of 6 and 7 respectively. So they are very strong binders of these of HSP90 in this screen. And it's exactly what we found when we either overexpressed PSKH2 with strep tags and did mass spectrometry, where we did this analysis. So this is an immunoprecipitation analysis where we've either expressed C-terminally tagged strep PSKH2 or N-terminally tagged strep PSKH2. And this band here is a non-specific band that our PSKH2 antibody recognizes, but as you can see, it does recognize these overexpressed proteins. Whoops, and we then pull them down with streptavidin, and in our control, we don't isolate any of the HSP90 complex, but PSKH2 tagged at the NLC terminus forms, we believe, a strong complex with HSP90, CDC37, and FKBP5. So that confirms our proteomics data. We also went back to look at precipitation of PSKH1 binding partners because of the history that was revealed by doing data and literature searches. And PSKH1, like PSKH2, a very understudied kinase, also binds to the FKBP CDC37 complexes. So in the last few minutes, I want to share with you new unpublished findings which show that the PSKH2 C-terminal tail is required for this chaperone binding complex. So we've made both wild type and this delta C320 version of PSKH2 and expressed it in 293 cells. Here's our input. And we then did an immunoprecipitation. And whilst the wild type IPs PSKH2, CDC37, and FKBP5, but you can see clearly if we take off this C-terminal region, we can IP the protein, we can IP the pseudokinase, but we no longer see interaction with the chaperone system. We can also disrupt chemically PSKH2 binding to HSP90 CDC37 with drugs like galdanomycin, which massively destabilize PSKH2. I hope you can see that as we titrate in the drug, we lose the PSKH2. Our positive control is the kinase AKT, and you can see that being degraded here. We can add, we can add solastrol, which modulates the HSP90 CDC37 interaction. And again, you can see degradation of PSKH2 very clearly here. And somewhat paradoxically, if we add 17-AAG, which is a related galdanomycin HSP90 inhibitor, we actually stabilize, PSKH2. And that's a really unexpected result, although we still degrade AKT. So we don't understand what that means, but we think this chemical data show that PSKH2 is clearly involved or is targeted as a client by the HSP90 CDC37 system. And we can rule out a couple of things. So this is an HSP70 inhibitor, this Ver compound, which doesn't destabilize PSKH2. And we can also show that dexamethasone, which increases FKBP5, which we believe is a bona fide target of the pseudokinase, doesn't have any effect on PSKH2. So just to end the talk, I know what's interesting to many people who work in the pseudokinase field is to ask, is this pseudokinase actually have any properties that allow it to bind to ATP or even to undergo non-canonical phosphorylation of substrates? And to start answering those questions, we've generated PSKH2 where we've put back in or knocked back in the catalytic asp or taken out the ATP binding lysine or the magnesium binding aspartate and expressed these in 293 cells. And you can see expression is good for all of these mutations. And none of those changes actually block the ability of either wild type, and compared to wild type, any of those mutations do not change interaction with the HSP90 system. And we think this is totally consistent with our finding that it's the C-terminal tail of PSKH2 that mediates those effects. So just to end, I mentioned that we found in our proteomics data that PSKH2 clearly binds to complex 1 in the mitochondrion. So that's made us to start think about where PSKH2 might be in cells. We believe, as I say, that it's myristylated. We are biochemically proving that at the moment for a paper that we're writing, but just want to share with you this unpublished data as well, which shows that PSKH2 co-localizes with the mitochondrial marker ATP5A in a mitochondrial Rix fraction, so that's M, and it's found a little bit in the cytosol as well. EGFP is only localized in the cytosol in our control. And if we delete the C-terminus of PSKH2, that C-terminal tail that binds to chaperones, we now really push all of the PSKH2 into the mitochondrial fraction under these conditions. That's really exciting because we think it might actually allow us to understand PSKH2 biology a bit better And the final thing that, of course, we want to know is, can PSKH2 interact productively or not with nucleotides? And to do that, we're using acyl ATP probes to detect binding to the potential lysine, which is conserved in PSKH2 in the beta-3 motif. And that data is currently being processed. So to conclude, PSKH2 is a client of the HSP90 CDC37 system. This works through the non-pseudokinase C tail, and it doesn't seem to require any of the classical amino acids that define PSKH2 as both a kinase and a pseudokinase. Expression is sensitive to HSP90 and CDC37 inhibitors, although we don't understand why some HSP90 inhibitors don't give this destabilization. It's found in a membrane fraction that's strongly enriched in mitochondria, and we believe this is probably through an N-terminal modification. But really, PSKH2 biology still remains a mystery, and the way we're going to get at this, we think, is to either prove or disprove that PSKH2 can bind to nucleotides, and then set up a chemical genetic method to try and understand PSKH2. And you know, PSKH2 has been a really tough protein to work on. It's the toughest pseudokinase we've ever tried to make, even though it's a small pseudokinase. And we'd love to find a way of making recombinant PSKH2. And we're currently obviously working out if the HSP90 system co-expressed is going to help us with that. Okay, so I will end my talk there. I'll stop sharing my screen. I hope that went okay. I actually couldn't see anything. And interestingly, my battery nearly ran out, and the dog went berserk behind me. I'm sorry for the dog barking that you would have heard. He's still going. But I'm ready for any questions if there's time for any.
Yeah, thanks a lot, Patrick. Beautiful talk. Would like to ask everybody listening in to post the questions in the chat. Maybe waiting for this, I have one question. So what struck me is in your schematic drawing of the protein, you put a calcium calmodulin binding domain that also carries the two phosphorylation sites that is usually found in active Kinase, and they are usually preventing calmodulin binding. So did you detect calmodulin, and if not, was the protein phosphorylated that you pulled down?
Yeah, that's an excellent question. So we have been unable to detect calcium calmodulin binding to PSKH2, but the residues that are found in PSKH1 are essentially identical in that C tail. I agree that the model with PSKH1 is that those autophosphorylation events then block calcium calmodulin binding, and there's only one paper that's ever done that, but we're planning to repeat those. It's actually quite challenging at the moment without having a good source of 32p ATP because of the pandemic, and this has actually slowed some of these experiments. But yeah, it's a great point. We cannot find calcium calmodulin associated with PSKH2, and that could actually be because there's another protein bound instead, or that those residues are non-functional in PSKH2. We have looked for it, and we can't see it either in cell preps or under biochemical conditions where we've added those proteins back to IPs. So, you know, maybe as it's become a pseudokinase in primates and it no longer requires catalytic activity, which, again, we haven't proven so far, but we suspect, there may be no requirement to bind calcium calmodulin either, because that's the cofactor is no longer needed. So we think those are interesting questions for the future.
Yeah. I mean, it is interesting because this could regulate pseudo substrate inhibition, because this domain would block the substrate binding site.
Yeah, so there's probably other proteins that bind there. That's absolutely right. So I'm just going to look in the chat. We've got some questions in the chat in the Q&A.
Yeah, go ahead, please.
So Tom asks about any genetic links to disease with PSKH2. I mean, obviously, I've scoured the databases, and you can find with the odd mutations or the odd expression analysis, yes. Nothing that jumps out at all, though. So I'm not going to oversell PSKH2 as an obvious oncogene or an obvious tumor suppressor. There's no evidence for that. But, you know, we're waiting again to have more data, and PSKH2 is often absent in some data sets for some reason. Greg asks, did you find the mitochondrial HSP90 trap 1 in the PSKH2 pull down? That's a great question. We haven't looked, and I need to go and look for that. The model we have at the moment actually is based on a few very minor experiments, is that this is a cytosolic HSP90, a CDC37 complex. But yeah, that's a really interesting thing to look for, and we'll have a look. Is there functional redundancy with PSKH1? Well, we predicted this many years ago, Elton, and we wrote various grants to try and prove it. And we don't see any interactions when we've made the proteins and mixed them together. Functionally, we haven't been able to do the experiment with siRNA because we haven't got a phenotype with PSKH2, and the Golgi fragmentation experiments with PSKH1 knockdown are challenging. You know, all the work that's been done on both these proteins has been done with overexpressed protein. There's no real good endogenous antibodies that prove that we're doing those experiments properly. But yeah, it's a good question, and I would say there probably isn't redundancy, but you know, Kanan may have a different opinion. If you look in genomes where there's no PSKH2, there's usually a PSKH1. So that might argue that there is. And certainly we believe that the myristylation, palmitylation sequences are probably functional in PSKH2. So if you don't have PSKH2, PSKH1 probably takes on some of those roles.
Yeah. So Pat, in the interest of time, perhaps there are other questions. Maybe you should answer it in the chat box. Probably move to the next speaker if that's okay with you. Thank you. Yeah, there are other questions, but maybe some of those we can cover in the panel discussion, or if you don't mind answering a few of them in the chat box. Okay. So, Stefan, do you want to introduce the next speaker?
Yeah, of course. So the next speaker is James Murphy. He's a professor and head of the inflammation division of the Walter and Eliza Hall Institute of Medical Research in Melbourne. And his lab has a long interest in protein interactions underpinning signal transduction. And he has a long-standing interest in pseudokinases, particularly the pseudokinase MLKL. And seeing the image, I think this is what he is talking today about. Yeah, I'm looking forward to your talk, James.
Thanks so much, Stefan, and thanks also to Canon for the invitation to talk here today. I guess it's quite appropriate to talk about something so grim because, you know, it's after midnight here in Australia. So, you know, the witching hour, I guess. I can report that everything's okay in the future. So I'm going to tell you about MLKL, and we've been working on this for, I think, coming up 15 years. So this has been a real hobby of ours, and I think we've come a long way. And what we've learned bit by bit is about its potential function in disease and how it might be an attractive therapeutic target. So we've already heard about this already from Pat about what a pseudokinase is. Of course, it's something that looks like a protein kinase, but it lacks these one or more of these key catalytic motifs, and therefore it's either dead or it has a pathetic catalytic activity. And so I like to think of them as protein interaction modules. And it also raises the intriguing question of, well, perhaps maybe active protein kinases also could be considered this way. But for so many years, we've been blinded by their catalytic functions that we've missed this opportunity to understand this prospect. And the other thing about it, which has already been covered, is that these things are, the thing that unites them is their dissimilarity to other kinases. So they aren't really a family, they're a collection of outcasts. And we've already heard about the 60 odd that are in rodent and human kinomes, but also about the expansion in some organisms like Toxoplasma have about a third of their kinome predicted to be dead kinases. And Pat, me and others have written all sorts of things really vibing on this idea of zombie proteins, which has been a good deal of fun over the years. More recently, Peter Mace and I put together some thoughts on where we're at the moment about pseudokinase function. And these are what we've distilled down over the years, what the four major functions are. And we've already heard a bit about we're depicting a pseudokinase in gray here, but the principal role really is the best characterized examples are as allosteric activators or perhaps even negative regulators. So on the right here, another common mode that they serve is enucleating the assembly of signaling complexes, so acting as signaling hubs. Less common is this idea of signaling switches or integrators of signaling. And here on the right down the bottom is probably the unicorn of examples of what they might do, which is really as either competitive inhibitors where they can poison assembly of hollow enzymes, or they can sometimes act as substrate sinks. But there are very few examples known of this. But what I'm going to tell you about today is what we've learned about MLKL which in particular takes a signal from an upstream kinase RIP3, and this converts it into a different form. And now, after many years, we've got some very solid evidence of this, which I'm going to show you a little later on. So MLKL, until very recently, when we started working on it, in fact, we didn't know what it did. It was a protein of unknown function. And you can kind of tell from its full name, mixed lineage kinase domain, like that when it was named, no one knew what it did either. And it was only really in 2012 that it was implicated in a very topical cell death pathway program, necrosis or necroptosis, which I'm going to tell you a bit more about in a second. But when we started, all we knew about it was it had an N-terminal domain, as we imaginatively named it. And it turns out from our work and others that this is the domain that's important for membrane permeabilization, which really underlies MLKL's lytic function. But the pseudokinase domain serves as a regulatory domain, and we knew it was catalytically dead because it lacked two of the key catalytic motifs. But our studies went on to show that it could bind ATP, and more to the point, ATP competitive inhibitors. So there's some work by Cardia Pierotti, which was published last year in collaboration with Guillaume Lassen and many others, where we found a compound that could bind to MLKL's pseudoactive site. But we kind of got the surprise that its upstream regulators, the RIP1 kinase and RIP3 kinase, were also targets of this compound. So I wanted to mention this because this raises a couple of interesting points. So one is that because the ancestors of pseudokinases often share similarities with conventional protein kinases, there may be some difficulties in selectively targeting their pseudoactive sites. And the other thing is something that Pat really covered already, which is that what if existing drugs, including all the FDA-approved drugs out there, what if they have a kinase target, but also pseudokinase targets? And that's something that we haven't been looking for. So that's something which commonly is in our thinking, particularly because we can use some of these drugs as leads to potentially target pseudokinases through some med chem. But I'm not going to dwell any further on this. What I'm going to tell you about is over the past year, we've unfurled a couple of 4 checkpoints in MLKL's regulation. And we use some, I think, some really cool tools to do this. And that's what I want to share with you today. But firstly, necroptosis. So these are the 2 papers that led to its MLKL's implication in this pathway in 2012. And necroptosis, as you can imagine, is so named in order to remind you of apoptosis, but it's a necrotic pathway. So apoptosis, for those of you who are unfamiliar, is when a cell will shrink and the membrane starts to bleb, and then another scavenger cell will come and eat it up. So this is designed to be a rather clean exit for cells and often used in development, whereas necroptosis doesn't use these proteolytic enzymes, the caspases. Instead, what we've got is a permeabilization event that occurs following some sort of insult, which leads to the cell swelling and rupturing and releasing these alarmins or damps, which elicit immune reactions. And the key point here is that MLKL is the terminal effector in this pathway. And the other key point is that it's designed to make a mess. It's designed to be inflammatory. And this is something that has been cataloged in a number of mostly animal models, but some human studies as well, which I've summarized here on this poor person. But the idea being that errant necroptosis is really the underpinning of a number of inflammatory diseases, and we're interested in targeting a number of these by targeting the necroptosis pathway. And also, its origins lie in ancestral host defense. So it started as an immune pathway, but now we think of it as being dysregulated in human disease. So this is our up-to-the-minute thinking about the pathway. So I mentioned that over the past year or so, we've identified 4 new checkpoints in MLKL's regulation. And so I've numbered these down here. And what I'm going to do is walk you through the evidence for these findings in a rather concise way, because much of this work is published. So in human cells, we found that RIP3, the upstream kinase, binds to MLKL in this dormant form. So this is obviously really important to restrain MLKL in an inactive form in our cells, because we don't want our cells running off and dying of necroptosis and eliciting immune reactions in our healthy cells. But when cells receive cues from death receptors or bacterial, like pathogen detectors. So in our case, we usually use TNF in the lab to stimulate the pathway. And in certain cellular environments where caspase 8 and E3 ligases of the IAP family are diminished or inhibited, then there's a cue that we don't fully understand yet of MLKL being recruited to this cellular platform called the necrosome, which is a megadalton cytoplasmic complex, which has the upstream kinases RIP1 and RIP3 in the complex, emit a number of other proteins. So in this complex, RIP3 can phosphorylate MLKL, and some data I'll show you that we've recently generated indicates that this causes a structural interconversion, and this accompanies a release of MLKL from the necroptosis, from the necrosome, which enables it to proceed to kill cells. And this relies on active trafficking of the protein to the plasma membrane, and there is a threshold amount required before membrane disruption can occur. And so what we wanted to do was study this in human cells, and we needed to generate a number of tools in order to do this. And we really wanted to focus on endogenous proteins because we didn't want to get carried away with effects we might see through overexpression. So what we did was turned to the Coidi lab at NYU, and they've been wonderful collaborators for us. And they're experts in generating monobodies, which are fibronectin 3-like domains, very compact domains, and they've generated libraries with these residues that have been randomized, and so we could screen for ligands, synthetic ligands of this nature, which are really convenient because not only for structural studies, which I'll show you, but also because when we express them, they reside in the cytoplasm. It can be very useful for picking apart the signaling events. So we generated baits, so for each domain in MLKL, and we were able to generate some wonderful monobodies that have nanomolar affinity for each of the respective parts of human MLKL. And firstly, I'm going to tell you about two of them which bind to the pseudokinase domain of MLKL. This is a study that we recently published. So firstly, monobody 32. So this is an interesting one in that we could pick out MLKL basically in any way, shape or form within the cell. And this was integral to us identifying this dormant MLKL complex. And we could rationalize this structurally. This work done by Meng, a PhD student in the lab, who identified this monobody binding to the hinge of the MLKL pseudokinase domain. And you can see that there's an activation loop helix here, which pushes away the alpha C helix. And so we proposed on this basis that this open conformation, which resembles an inactive conformer of conventional protein kinases, likely represents the inactive form of MLKL, the dormant form that's sitting in our healthy cells. So we also, in the course of that screening endeavor, identified another monobody, monobody 27. And this is a really cool tool because it enabled us to pick out this other second step in the checkpoint in the pathway, which, so in resting cells, we couldn't detect MLKL with monobody 27, but after we applied a necroptotic stimulus, MLKL disengages from RIP3 and is then, the epitope is then visible for monobody 27 to bind. And this is something that Sarah, another PhD student in the lab, figured out. So you can see here that monobody 32 binds rain or shine to MLKL and also brings down RIP3 and IPs, whereas 27 only binds MLKL once the cells have experienced a necroptotic stimulus, and that corresponds with phosphorylation of MLKL, but we don't see RIP3 in the IP. And so this was our thinking, that we needed RIP3 to MLKL to disengage from RIP3 in order for monobody 27 to recognize it. And that's something that Sarah could verify biochemically in RIP3 knockout cells, where now she was able to IP MLKL regardless of whether there was necroptotic stimulus or not. And again, this is something that Meng was able to rationalize with another structure. This time, the monobody binds at the Pac-Man aperture here, where ATP would bind, and the MLKL is in a closed conformation with without an activation loop helix. And if we superimpose this on the mouse RIP3 MLKL structure, you can see that there's a bit of overlap between where the monobody binds and the interface between these two proteins. So this really argues for some competition between the two, such that we require MLKL to break away from the necrosome in order to be recognized by monobody 27. So this is super cool because now we've got a probe that can detect activated MLKL, aside from verifying this disengagement step as a necessary checkpoint. And this is something that we further tried to rationalize with the help of Wayland and Cannon using molecular dynamics. So our initial thinking was, on reflection, probably quite simplistic, and that was phosphorylation of this activation loop, the two substrates here of RIP3, would that lead to unfurling of this activation loop helix? And it turns out, if anything, it probably stabilizes the helix more in these MD simulations. So back to the drawing board. And so then we came up with this idea that, in fact, this transition from this open form to this closed form, so the open being what we think the inactive form is to this closed form, which is the activated form of MLKL, the barrier could be, the energy barrier could be diminished by phosphorylation. So that's in keeping with the idea that this phosphorylation event accompanies this dissociation of MLKL from the necrosome. So, but we didn't stop there, of course. We had other monobodies that bound to the N-terminal domain, the killer domain MLKL, and we found that these were able to identify another checkpoint, which was a trafficking checkpoint, in which MLKL is actively trafficked to the plasma membrane where it does its damage. So initially, ever so briefly, because this also is published, and this is work mostly by Emma Petrie when she was in the lab, but in human cells, if we express some of these N-terminal binders in these two human cells, and we expose the cells to a necroptotic stimulus, you can see when the monobody is present, we protect the cells from death. And that occurs in two human cell types, but it doesn't occur in this mouse cell line, and it didn't occur for our monobody 32, which binds to this pseudokinase domain, which I told you about earlier. So this tells us we've got a really selective reagent for human MLKL and a really efficient way of blocking cell death, which was very cool. But we wanted to figure out how it was working. And the most obvious thing could have been that it was going to block MLKL phosphorylation, and clearly it has no impact on MLKL phosphorylation relative to our control, which doesn't block cell death. It also had no impact on the monobody's ability to bind to phospho-MLKL and these IPs. But this work done by Emma and Sarah again, but Catherine Davies found that, in fact, it was not a ligamerization in these blue native page gels, but a membrane translocation that was blocked by this monobody. So normally, this is a fractionation to cytoplasmic and membrane fractions, and up here a blue native page. So under basal conditions, MLKL resides in this lower molecular weight species, and it's only in the cytoplasm, but following a necroptotic stimulus, it migrates into the membrane fraction and forms this high molecular weight species. In the presence of the monobody, we get this high molecular weight species, but it doesn't go to the membrane. And we could figure out why this was by solving structure with the help of Rich Birkinshaw and Pete Savitar, and we could see that monobody 33 binds to the alpha 4 helix of the 4 helix bundle, and we knew that wasn't perturbing lipid binding or membrane engagement because those key residues are on the other face. But we did a mutational analysis and we found a number of key residues shown in red here, and what we concluded from this was that this site is essential for engaging a trafficking chaperone, although we don't know the identity of that chaperone yet. It is something that we're currently pursuing. Just a brief detour into the last step of what I showed you, which was the threshold step. So we developed a number of tools in-house, as well as optimizing commercially available reagents. So these are antibodies, monoclonal antibodies. And really the genesis of this project was an observation that Andre Sampson and others in the literature have made, that phospho-MLKL occurs much earlier. So this is the hallmark of pathway activation, occurs much earlier, quantified in blue here, than cell death, quantified in red. And he wanted to figure out what the temporal gap was. And it turns out it's a spatiotemporal gap. So he used imaging to identify what he saw as MLKL clusters in the cytoplasm, which we think correspond to necrosomes. And he also saw these phospho-MLKL, so activated MLKL at the plasma membrane. And this is something that he, with the help of Locke, was able to quantify. So we can see here that the size of these clusters grows, but in the plasma membrane with time, following stimulation with a necroptotic stimulus. And in the case of these phospho MLKL hotspots at the plasma membrane, they grow in size and intensity with time. And the kinetics really correspond with when we start to see death. He also figured out that this was relied on a trafficking pathway through use of inhibitors. So he found that a combination of Golgi, microtubule, and actin trafficking inhibitors was really comprehensively blocked cell death. And he also found that he had added at a really late stage post-stimulation. So he could halt the killing. And that really gave us this idea around this threshold need to be met. So just to summarize, so what we've found are these 4 new signaling checkpoints, which really revolve around the dormant form of MLKL, which is recruited to the necrosome, where it can be activated by phosphorylation by RIP3. And this leads to a structural interconversion that accompanies its dissociation from the necrosome, which then allows MLKL to be trafficked by either chaperones or this microtubule actin and Golgi pathway to the plasma membrane, where it accumulates. And once it hits this threshold, it's able to permeabilize cells. And so in the course of doing this, we've generated a number of cool tools, which I'm really excited about. And we have also got the first formal evidence for this idea that the protein serves as a molecular switch. But we don't know everything, of course, which is lucky because it's really interesting. So we don't know what the identities of the chaperones for membrane translocation are. So we don't know exactly what's happening here. And we certainly don't know exactly what's happening in the membrane and whether there might exist other co-effectors. So lastly, thank people as I've gone along, but Sarah and Meng did the monobody 27, monobody 32 study that we recently published. Meng and Catherine have recently solved the structure of the RIP3 MLKL complex, which I didn't have time to talk about, but it's super cool, and hopefully it'll be in the public domain soon. Andre with the imaging, ably supported throughout all of these projects by Cherie and Sam and other members of the lab, and Emma, who kicked off the monobody study with the COE lab, who we're indebted to for these reagents. Pete and Rich for structural help, and a number of other collaborators, both at WeHi and externally, and also our funding. So thank you.
Yeah, thanks a lot, James, for this really nice talk. Again, please post your questions in the chat. How are you validating your monobodies or your binders once they are produced? This is just on target, or you test them in cells like in IP assays?
Yeah, both really. So we've been fortunate to be able to crystallize a lot of them with their target, and it certainly rationalizes why they'd bind specifically. But we thought that all block signaling, so it was a bit disappointing when those pseudokinase binders didn't. But it turned out that it was probably more exciting in the end, because we were able to identify some different forms of MLKL through their complexes. But yeah, mostly through cellular studies as well as the biochemistry.
Okay, you have the first questions coming up. Do you see that in the chat, or shall I read it to you?
The Q&A is one.
Yeah.
So,
yeah.
Yeah, can RIPK3 phosphorylate MLK by cycling after disengagement is the question.
Yeah, we don't really know. It turns out, if you try to do these things in vitro, I don't think you fully replicate what you see in the cell, because I showed the kinase domains, but there are other domains as well. So there are other scaffolding events. So I don't think we fully recapitulate that at the moment. We kind of think it must, but this is a complex with a lot of RIP3s present, so we can't really say. So in cells at least, but if we do take data from longer time points, then we start to see D-phospho-MLKL coming down too. No, phospho-MLKL comes down too, so that means that it must be forming a bigger complex and bringing down some of the already phosphorylated material. So I would think it is quite recessive, but I'm not sure how we'd prove it. Yeah, there's another question. Do you want me just to go for it?
Mm-hmm.
So from Adil, there's a question about whether the temporal gap between MLKL phosphorylation and death is consistent between cell types and stimuli. No, it's not. So HT29s, which are a workhorse, have quite a small nucleus relative to their cytoplasm. So there is a lot of trafficking required. But if you take a cell with different morphology that looks more like a trimmed up fried egg, then there's less of a gap between trafficking and less of a requirement, it seems, for assistance to get from cytoplasm to the plasma membrane. And we often use a bunch of different stimuli, and some of them tickle the pathway, some of them just smoke it. So that is another factor. I've got a question from Diep. Is it correct that the pseudokinase monobodies can't rescue cell death? And if so, why? Yeah, we don't really know why. We think it probably relates to the fact that RIP3 and MLKL preform a complex, maybe co-translationally. And so to get a wedge in there with a monobody might be quite difficult, particularly since some of them are only overlapping in terms of the epitope they bind. I would have thought, less than 100 nanomolar affinity would have been enough to get in there and disrupt it. But yeah, we don't really know the answer why. There's a question there for you, Canon and Wayland.
Yeah. So that's a question for us. So how long are the MD simulations? I think they were microsecond timescale simulations, Katrina. Yeah, that's relatively short to probe these conformational transitions, but Wayland did multiple microsecond simulations, starting from different snapshots, and so we were able to kind of get an idea of the free energy landscape using that. So yeah, thank you so much, James, for that wonderful talk, and thanks for giving this late at night. I know it's kind of difficult, but really appreciate it. That was great. So I think this is a good segue to the next talk here, where Ben Major is going to tell us about how he's developing new tools and approaches to probe the non-catalytic functions of dark kinases. And as you've already seen from Pat's and James's talks, that the pseudokinases, one of their main functions is to really scaffold and mediate protein-protein interaction functions. And so Ben is a professor of cell biology and physiology at Washington University in St. Louis, and he's going to tell us about protein-protein proximity networks of understudied kinases using mass spec approaches. So Ben, if you want to share your screen.
Hello, everyone. Can you see the presenter view?
Yes.
I hope.
Yes, we can. Good.
Great. And laser pointer. So thank you, Kanan, and thank you all who I cannot see but appreciate your presence. I'm going to share with you a data production pipeline project that we've been involved in for the last 4 years or so, and that is part of the IDG program. My lab within the kinome-centric IDG group is charged with defining protein-protein proximity networks and protein-protein interaction networks for the understudied kinome. And to do this, we're expressing fusion proteins in cells, where we take an understudied kinase and we tag it with an epitope tag for traditional affinity pull-down mass spectrometry, and with promiscuous biotin ligase. The idea being that, and both platforms will use mass spectrometry as our readout. You know, quite simply, the idea is that if we can capture, identify, and quantify the proteins that bind understudied kinases, understudied or studied pseudokinases, and we know something about those proteins, where they live in the cell, their disease association, their functions, we can infer or at least hypothesize the function of the kinase. So I said we're taking two approaches. One is the traditional IP mass spec that will reveal direct protein-protein interactions and bridged protein-protein-protein interactions. And then employing newer technology with turbo ID, mini turbo ID. This is the promiscuous biotin ligase. You feed your cells expressing this fusion protein, biotin. That biotin is activated and covalently attacks lysine residues on the surface of proximal proteins. So we're taking this combined approach to map protein-protein interaction and proximity networks in order to illuminate these understudied kinases and pseudokinases. The IDG program has defined the kinome and the understudied portion of the kinome to be today at about 150 kinases. So we've been looking at these 150 and asking some informatic questions with respect to their darkness, and specifically respect to how dark they are with respect to protein-protein interactions. So 55 of which have never been used as a bait, and 17 have never been seen as a prey with another bait. So there's a dozen or so, I'm forgetting the exact number, that have never been seen in a mass spec-based interactome study. So they're quite dark. With respect to pseudokinases, 20 of the 150 understudied kinases are pseudokinases. And I'm going to walk you through, we've to date hit about 65 of these dark kinases we've developed networks for, but only 2 of those are pseudokinases. So, but over the next year to year and a half, we will get closer and closer to completing this set of 150. So our pipeline looks like this. So our step one is to get the open reading frame, and we've pulled these from publicly available clone sets. They're all dark, meaning that many of these were not in these libraries. So to that end, we've been collaborating with SGC, as well as just cloning open reading frames out. Once we have those, we sequence validate, and then we're using a multi-component gateway system to recombine our open reading frames into new destination vectors that we've created that have either an N-terminal tag or a C-terminal tag with the mini turbo cassette and a flag cassette. We've done a lot of optimization around this pipeline that I'm not going to go into in great detail. But one of the things to be very cautious about is overexpression artifact, particularly with respect to biotin proximity analyses. So we moved to, instead of a strong promoter like CMV, we moved to the ubiquitin promoter to drop the expression, and that seemed to help a lot. And then we create lentiviruses, stable cell lines, and validate by Western just that the biotinylation reaction is working, that our beta is expressed, it's the right size. And oftentimes we'll do a time course to figure out the appropriate amount of time post-biotin before we scale up and move to mass spectrometry. So we're kind of finishing, we're about halfway done, maybe a little more halfway done in producing the data. So our efforts now are moving more and more into informatic analyses of the networks. What does all of this data mean? With respect to function and subcellular localization, disease associations, substrate identity. We are, so the 0.5, so as a part of the IDG, we are releasing these data in real time or close to real time. So I think we've got up on the Dark Kinome Knowledge Base, dkk.org. You could go there and see the networks that we have defined already and have released. And then, we're, as I mentioned from the informatics, we're searching for mechanism and phenotypes and disease relevance. So to date, we've cloned 111 of the open reading frames. We've moved 93 of those into the Lenti system with this multi-component gateway. We have 81 cell lines created, well, 81 baits and cell lines out of 160 total, and 61 so far have done. A portion of these have been moved, have been publicly released. So I want to spend a quick minute on what has been a challenge that maybe if you're not in this space, you don't think of it often enough, but how do we take the data that's coming off of the mass spec and score it, probabilistically score it for true positives and false positives? We've had a challenge here because these are proximity networks and not protein-protein interaction networks. So this is an example of what we get. So if we use CDK2, which is a very well studied and understood cyclin-dependent kinase, using this mini turbo system, we get 3,300 proteins as proximal. And then we use a mixture model-based approach from Alexei Nishevsky's lab called SAINT. And with SAINT, and this is all versus controls. We have a number of controls built in to the model. So we then get down to 709 proteins, which is still, it's a lot, and which isn't to say they're not real, because again, these are proximal protein networks. But for today to reduce somewhat, we use Compass, which is more of a heuristic scoring approach that will point to uniqueness across our full set of baits. So drawing an arbitrary threshold within Compass, you can see that our high confidence network of 35 proteins proximal to CDK2, and in red are those proteins that have been previously reported to be CDK2 associated proteins. So we're enriching for what is known. Here's an example of an understudied kinase, ADCK1, 2,690 proteins, SAINT down to 535, sorry. And then with Compass, we get 25 proteins. It's understudied. There are no known protein interactions. And we have this network. So one of the things that's easier to do, so far as it's plug and play, is to take this network and ask subcellular localization questions. The vast majority of these proteins are nuclear, which was a little bit of a surprise because ADCK1 is classically from Drosophila studies reported to have mitochondrial biology, but we see it associating with nuclear proteins. Stepping back and looking at our network version 1.0 so far of 61 baits, this is the distribution of the number of prey per bait, and we can just look at the 0.9. This is the SAINT scored probabilistic networks. So you can see that we've got some baits that the networks are small. Relatively fewer are upwards of 500 or so prey proximal proteins. I lost my ability to advance. We'll do it this way. Oops. Okay, I'm back. So if we distribute across in this visualization, you can see we only have 4 baits with 1,000 prey. And I put this up to highlight to you that we're doing some time course studies of 30, 60, 120 minutes post biotin, and expectedly at the shorter 30-minute time point, we're capturing smaller proximal networks. It's exactly what you would predict. And that at the longer 120, that's where we start to see greater number of prey proteins. So once the data, if you go to DKK and you start to look at these data, you can pick your time point and how short of a snapshot in time you want to ascertain the proximal proteins. The 2 pseudokinases we've looked at thus far, it's CAMK5 and TBCK. See pretty big differences in proximal protein networks. We haven't started to study in depth these networks, but I can say that gene set enrichment type of analysis for the CAMK5 network points to guanine nucleotide exchange factors, plasma membrane localization, Rho signaling, which The little that's known of CAMK5 is that it's involved in neural tissues and dendritic spine maturation. So at least biologically, functionally, there may be a connection there. And we see the one known interaction for CAMK5. So I'm going to finish with more of a deep dive on 3 kinases. And I appreciate this is a pseudokinase symposium we have here. But absent pseudokinase data, I thought I would give you an idea of the types of things that we do from the data that we generate with these networks. So this is with the casein kinase 1 gamma family. These are understudied members of this CK1 family. They're serine threonine protein kinases. Here they are on the tree, related to CK1 alpha, delta, and epsilon, more widely studied. There are 9 publications thus far on the gamma family, and it's kind of all over the place. So we just heard about RIPK3 signaling and necroptosis. CK1 gamma 1 and 3 have been shown to promote that most recently. Also involved in immune signaling via RIG-I and the TLR receptor, DNA damage response. There's a couple of papers that suggest that it's involved in Wnt beta-catenin signaling, and you'll see that that is where we have taken this story. Weak disease connections overall. And there's at least 2 tool compounds available, and we will use both of them. This I didn't have them on the front screen, but our tool compound work has been really made possible by Allison Axman and Tim Wilson within Structural Genomics Consortium. So for all of these kinases, I just walked you through the pipeline of data production around the networks. But the other thing that we're doing is functionally ascertaining how kinase overexpression impacts signaling pathways. Can we bin understudied kinase and pseudokinases into signaling pathways? And to do this, we're using engineered transcriptional reporters that have 12 copies of a downstream pathway transcription factor. So here you can see that if you just focus here for the Wnt beta-catenin reporter, here's our control. If we overexpress beta-catenin, we activate the reporter. We overexpress CK1 gamma 1, gamma 2, or gamma 3. Gamma 3 uniquely activates the Wnt reporter. With the Notch reporter, again, positive control of the intracellular domain of Notch, the NICD, activates the reporter. Gamma 3 somewhat activates it. NF-kappa B, there's a little bit of activation across the board. TGF-beta, nothing really. NRF2, oxidative stress response, nothing. And neither really with retinoic acid. So this sort of a functional evaluation, and this is not all of the reporters, point us immediately for CK1 gamma 3 to the Wnt pathway. And then we look at the protein-protein interaction network and protein proximity networks, and we ask questions about pathways, right? So the proteins on the left that associate with gamma 3 are all known to be involved at one level or another with the Wnt pathway. So we had a handle, both physical and functional. And then we go in and we start validating. So we can see that gamma 3 and gamma 2, for the most part, is proximal. This is a proximal assay to the Wnt receptor LRP6. Moving a little bit more mechanistically, I want to give you a background on how the Wnt pathway works real fast. So in the absence of a Wnt ligand, beta-catenin is the transcriptional coactivator. This beta-catenin is phosphorylated by this complex of proteins. Phosphorylated beta-catenin is ubiquitinated by a CUL1 scaffolded E3 ubiquitin ligase, and then degraded. So this is how the cell keeps the Wnt pathway off. Following Wnt activation or Wnt ligand presentation, you pull together the frizzled receptor and an LRP co-receptor. That results in phosphorylation of LRP6. That phosphorylation event is driven by GSK3 beta and CK1, and the literature is a little bit unclear as to which CK1 family member, if it's epsilon or if it's gamma, and then if it's gamma, whether it's 1, 2, or 3. That reorganizes these proteins. Beta-catenin is stabilized. It goes into the nucleus to drive transcription to govern a host of cell biological responses. So again, I already showed you this, but I'm going to show it again because it's beautiful. if we overexpress CK1 gamma 1 and look at luciferase downstream of beta-catenin, there's really no effect. If we overexpress gamma 2, not really an effect, but gamma 3 overexpression again gives this very unique, at least within the family, activation of the Wnt signaling pathway. And I'm realizing as I'm talking right now that I didn't show you, an alignment between gamma 1, 2, and 3 homology. They're highly homologous, 85% identical within the kinase domain. And overall structural layout of the protein and their domains are more or less identical, with the sole exception that gamma 3 has a 35 amino acid insertion within the C terminus of unknown function. I mentioned it was speculated that CK1 gamma phosphorylates LRP6, and we have antibodies against the GSK phosphorylation site and the CK1 phosphorylation site, and those are shown here. So if you treat cells with Wnt, LRP6 is phosphorylated, and you can see it's faint on your screen probably. That GSK phosphosite goes up, CK1 phosphosite is more or less the same. And then if we overexpress gamma 1, gamma 2, or gamma 3, we see a fairly robust induction for both the CK1 site and the GSK3 site. Okay, so now we move to some functional questions. If we make kinase dead CK1 gamma 1, does that abrogate its ability to phosphorylate LRP6 and activate Wnt? And you can see with the kinase dead version of CK1 gamma 3, it no longer activates Wnt. Here's the wild type, and then there's the mutant. We can do the same thing with live cell imaging. So here, Wnt beta-catenin drives GFP. And so wild type overexpression of CK1 gamma 3 in the blue line activates the reporter. And then if we make the kinase dead in red, there's a much suppressed activation. And this is both in the presence of Wnt ligand. If we look in the absence of Wnt ligand, the dotted line, there's no activation. So kinase activity is required. In a loss of function setting, this is where it's gotten more subtle, but we're starting to believe it, that if you knock out gamma 3 or gamma 1, 2, and 3, what effect does this have on the Wnt pathway? So you can see here's our reporter again, treat cells with Wnt, the reporter goes up. Knock out gamma 3, there's a statistically significant reduction. It's subtle, but it's there and real. If you knock out all 3, you have an exacerbated reduction, suggesting some potential involvement of the other players, functional redundancy. And then I also told you that if you treat cells with Wnt, the beta-catenin is stabilized. So here we treat with Wnt and do a Western blot for beta-catenin, its levels go up. And then we did the same thing in cells lacking CK1 gamma 3 or the whole family member. We still see the beta-catenin stabilization, but it's muted. And we're quantifying this. This is unpublished data, but we've seen it over replicate experiments thus far. And finally, again, working with Allison Axman and Tim Wilson, we've been moving into looking at probe compound inhibitors of CK1. And we started with this bottom compound that was part of the SGC library, the original PKIS library of kinase selective inhibitors. But we've more recently moved to this compound, which was originally published and reported by Amgen as a CK1 inhibitor. We've done some initial DiscoverX profiling. Both compounds are, I guess, reasonably selective. They're hitting about 14 to 17 kinases at less than 10% in vitro using 1 micromolar of drug, and this is their distribution. Why we moved away from the SGC compound is because they're hitting GSK3 beta and GSK3 alpha. These 2 proteins are, if you knock out or inhibit GSK3 beta, the Wnt pathway is maximally activated, right? This kinase is required for beta-catenin destruction. So we needed a compound that did not touch GSK3 alpha and beta, and that's why we moved to the Amgen compound. This compound is more potent against CK1 gamma 1, 2, and 3 versus the PKIS compound. And this is preliminary, but we have been able to show that if we overexpress gamma-3, we activate Wnt, and then in a dose-dependent fashion, we can suppress that activation with the kinase inhibitor. Ultimately, to show that this is working the way we think it is, we need to show that phosphorylation of LRP6, which we hypothesize is via CK1 gamma-3, that is suppressed by this inhibitor. So if you look here at the laser pointer, we have this induction of LRP6 phosphorylation following Wnt. In the presence of the drug, we do not. We need to quantify this again. We've seen it a couple of times, but we need it quantified. But we do, it makes us feel good to see this muted beta-catenin stabilization. And this is what I showed you before with the loss of function. So I think that CK1 gamma-3 is involved, but absent its activity or presence, you still can get a functional Wnt response with Wnt ligand because of data like this, that we're still getting beta-catenin stabilization and the reporter is still being activated. I'll go through this slide quickly because I think I'm running out of time. That we've been, with this mini turbo system, you can feed cells biotin to trigger your biotinylation reaction. And then you can fix your cells and come in, and not with an antibody, but with fluorescently labeled streptavidin to ask where in the cell is that biotinylation reaction happening as you know, as a surrogate in some ways, or a complement to doing immunofluorescence. And when we do this for CK1 gamma, we see that we're lighting up the cell membrane, which is where these kinases are thought to live. They're myristylated. So we can visualize where in the cell the biotinylation reaction is happening. And so, you know, so I think one intent of my talk today for you is so that you're aware that we're producing these data, both from the data production pipeline optimization. We're happy to talk with anybody if you want to get into this technology. And then you can go to the dkk.org and look at the data that we've produced. And, you know, every 3 months or so, we put another data set up there. And finally, I'll thank my lab. Dhaval has been doing most of the data production around the mini turbo system. Megan recently graduated, and she's the Wnt biologist that's studying CK1 gamma 3. And then Chase has been helping Dhaval in some ways developing these networks. And Danny's been a super cloning machine type of person. And I'll also thank, you know, Dennis and Smiranda, who've helped a lot with the informatics with Saint and Compass, and with beginning to informatically study these networks. And then the larger IDG dark kinome team. And with that, I'll take any questions that you have.
Thanks, Ben. Thanks for the fascinating talk. So while we're waiting for questions, you know, maybe I'll get started on a couple of questions that I had. So it'd be great if you could elaborate a little bit on how you're really, you know, teasing out the true positives and, you know, and the true the false positives and false negatives based on your proximity networks. If we could elaborate on that, that'll be great. And the second question was with regard to your CK1 gamma, you know, active site mutant. You've essentially generated a pseudokinase version of that. So how does it impact, you know, so of course, catalytic activity is lost, but presumably the protein-protein interaction functions are still maintained, right? So have you looked into that, and could you elaborate on that?
Yeah, both great questions. The latter question, we need to do the experiment. We have not defined networks for kinase dead. But you're exactly right. You know, with other kinase stories, we've done this in phosphoproteomics for the BRSK kinases. We see differential phosphosites and protein interaction networks of wild type versus dead. And it's a good idea, a good experiment. The first question you asked was with respect to the scoring. So, you know, Saint as mixture model is basically looking for enrichment versus your set of controls. That I think conceptually works well for protein-protein interactions for a flag pull down. But once you move into a proximal state, it's not designed for proximal scoring. But, you know, working with people like you and others, we may be able to define a better way to do it, but absent that, we're using Saint as our first pass thresholding. So we're just taking, Saint score of 0.9, just like a 10% false discovery rate. And then from that list, we are rank ordering based on uniqueness using Compass. So when you are interested in this, if you look at the data, do not trust at all, a line across the data that we or somebody else would draw, because there's meaningful stuff below that line. Yep. It's just how much data can you handle for the type of experiment and question that you're asking.
Okay, great. A couple of questions in the chat box. I can read it out for you, or if you're able to read it and answer them, that'll be great. So I think the
I can see. Yeah. So Alexandria asked a question that we're still defining. So,
if you don't mind reading the question, Ben, because
I will do that. Yeah. So she asked of the overlap obtained between a traditional IP-mass spec and the proximal data. So I think this has been a surprise to me, insofar as that the overlap is almost always smaller than we would have thought it to be. And I don't know if the community really understands why the overlap is appreciably less than I would have predicted going into this technology. Certainly, proximal networks are generally larger than the IP-mass spec, but well-validated, biological triplicates, multiple cell lines, N and C term tagged IP-mass spec data, you don't see it all in the proximal networks. And it depends on the bait. It's anywhere from 10% overlap to more than 50% overlap. But, we've seen this. I think Anne Claude's data has seen exactly the same thing. Mostly, that is the answer, but why that answer is there is, I think, a little more nuanced and going to be case specific. Please tell me when to stop for time.
Yeah,
I'm just going to
maybe a couple more questions, and then we can take a few of them at the end. Yeah.
There's a question with respect to LRP6, which is very well known within bone development and bone disease literature as a central component in the Wnt pathway. And the question is there any link to CK1 gamma to bone cells? Not that I'm aware of. The disease contribution, human pathology role for the CK1 gammas, I'm not clear there is a conclusion to mention to you here now. Most of it's all been cell-based signaling types of studies that I pointed to. Another question, do we account for changes in the protein expression linked to cell cycle phases? No, we do not. And particularly like some of we've analyzed all of the understudied cyclin-dependent kinases, and these were asynchronous growing stable cells in P15 plates at about 70% confluence. So we assume that and most of the data that I presented was all in HEC293 T cells. We've got a subset of it in some 159 breast cancer cells. So it's a great thought, but we would need a deeper dive and looking across 150 baits. Our goal one is just to get as many of them done as we can. I'll do one more.
Yeah. Mm-hmm. Yeah, you could answer that online. I think it's more.
Yeah, I'll answer those in the chat box.
Okay,
Thank
great.
you so much, everybody.
That was fantastic. So we move on to our last talk as part of this webinar, and that's from Michael Lazaris. He's an assistant professor in pharmaceutical sciences at the Mount Sinai School of Medicine. And he's going to tell us about the incredible ULKs, trying to understand the ULK family and the pseudokinase member within that. So I'll look forward to that. Michael, take it away.
Great, thank you. All right. Thanks so much for that, for the introduction, Kanan and Stefan, for the invitation to speak here. It's a great pleasure to be part of this pseudokinase community. So I'm going to tell you about some of our work today on this ULK family of proteins and sort of give you a background of how we got into it. And so the title is The Incredible Ulks and trying to understand the multiple roles, the sort of split personality of this ULK family between autophagy and neurological development. Okay, so initially I got interested in this pathway called autophagy, which many of you probably have heard about. So this is a highly conserved pathway in cells conserved from yeast to humans for breaking down cellular components all the way from individual proteins up to whole organelles as a response to starvation and also for quality control. And in yeast, this is where the pathway was discovered, and this is where they won the Nobel Prize for identifying all the components in this pathway. So there are a series of genes called Atg genes that were identified as being required for survival for starvation. And in yeast, there was a single kinase identified called ATG1 that is regulated by the yeast protein TOR, and that helps initiate this pathway of autophagy. And so if you look at other organisms, it becomes more complex once we get up to mammals. So in mammals, there's 5 proteins in this family that are related to ATG1, and these are called ULK1 through 4, and then STK36. And to give you an idea where this name comes from, it stands for UNC51-like kinase, and that's by way of the C. elegans protein UNC51, which was initially identified as having this phenotype of uncoordinated movement. So it had this sort of paralytic phenotype, and it was identified to be part of the same family. And so all members of this family have a N-terminal kinase domain, so pretty conserved kinase domain at the N-terminus, and then varying domains at the C-terminus of the proteins. And so ATG1 and these ULK1 and 2 proteins have a C-terminal domain that's involved in scaffolding the ULK1 complex, or the proteins involved in autophagy. And I won't talk about ULK3, that's been decently well characterized, but most of the talk will focus on this ULK4 protein, which again has this related kinase domain, in this case it's a pseudokinase, and then a series of heat repeats at the C-terminus, which are a series of protein-protein interaction domains that are found in a number of proteins, including mTOR. So initially, I was interested in this family of kinases because they're thought to be important therapeutic targets. So this pathway is thought to play a role in chemotherapy resistance and tumor growth. So many cancers are thought to be addicted to autophagy as an energy source. And ULK1 and 2 are sort of the main enzymes that are thought to be involved for initiating autophagy. So there's some redundancy between these proteins. ULK4, on the other hand, has this really interesting pleiotropic set of effects in different neuropsychiatric diseases and other diseases. And so there's definitely some interest from a sort of health point of view in understanding this protein. So I've told you that ULK1 and 2 are required for autophagy, but they also seem to have a neurological development role. So the Kundu lab showed that if you knock out both of them, it's not viable and eventually leads to death due to neuronal cell death. So if you knock out both 1 and 2, the neuronal cells are not able to develop correctly. And so we began our work initially focusing on ULK1. So this was work that I started at the end of my postdoc in Kevon's lab. And I was interested in this pathway, and that's how I got interested in the ULK family. And so we purified the kinase domain of ULK1. And because it was a normal kinase, we could do sort of pretty straightforward kinase assays. So just doing a p32 ATP assay, we were able to look for inhibitors, just looking at our in-house library, and initial screens identified nanomolar inhibitors. And I eventually did some medicinal chemistry to find more potent inhibitors. And I used these compounds to eventually crystallize the protein, including using some surface modification tricks to help it pack better, to obtain better crystals. And so we were able to solve the structure of ULK1. And this is that first initial compound that we found that binds into the active site of ULK1. No surprise, it's an ATP competitive inhibitor. And we're able to then do medicinal chemistry to make some more potent inhibitors against this kinase. Unfortunately, the challenge with kinases in general, it's very hard to get selectivity because of the conservation of the ATP pocket. And so our initial lead compound, which was a single-digit nanomolar inhibitor of ULK1, was actually quite promiscuous in the kinome. So in just a small panel, you can see it inhibits a lot of kinases. So we since developed slightly more selective ones, and a number of other groups have developed more selective compounds against ULK1. But that's sort of how, just to give you an overview of how we got interested in this family. And then, so in my own lab, I became interested in this protein ULK4. And to be honest, actually, I first caught my attention looking at when I was looking at this IDG project in the set of these sort of dark kinases. And I saw, oh, there's one in the ULK family. And I became interested in this protein. And from a biological standpoint, it has a number of intriguing features due to its relationship to a number of diseases. So one particular that caught my eye was its linkage to schizophrenia. So there were a number of genetic studies that found that there's a SNP for ULK4 that's linked to schizophrenia. And this is particularly interesting because it's actually a fairly widespread and devastating disease that's thought to be highly heritable, but there are not a lot of genes that have been identified for it. So anytime there are sort of new genes identified, that is intriguing because it might help shed light on the mechanism. And this same mutation is linked to autism and depression, which is often the case with these schizophrenia genes. And so early work on ULK4 showed that indeed it's important for development. So if you knock down ULK4, you get defects in cortex development in mice, particularly in axonal projection. And this sort of confirms this idea that in schizophrenia, although the symptoms usually manifest themselves later in life, the defects sort of occur early in development, from my understanding. But there are also several other diseases that have been linked to it. So actually just a few weeks ago, there was a new paper that showed a susceptibility locus for primary biliary cholangitis in the Japanese population and ULK4, and there are other associations, including acute aortic dissections, multiple myeloma. So really a pretty wide range of protein showing that has some important function in development and in human disease. So what does the protein do? And so because I'm a crystallographer, that was sort of our first instinct, was to start off to try to see what the protein looks like. And so we purified the pseudokinase domain, and basically using the same approach as for ULK1, we were able to obtain nice crystals of it, and we solved the structure, high resolution, and we actually crystallized it using that same compound that I showed you that we developed for ULK1 that was very potent towards this family, but not very selective, but it's very good in helping to crystallize the protein. And when we solved the structure, we sort of knew in advance, based on the sequence and alignments, that it was a really screwed up pseudokinase. But looking at the structure sort of brought home how messed up it was. So it has alterations in all 4 of the required kinase motifs, although the G loop is pretty close to normal. But otherwise, it's missing the DFG, the HRD motif. And then sort of most interestingly that you can see from this structure here is that instead of this lysine glutamate salt bridge in the active site of the protein, it has this leucine tryptophan hydrophobic bridge, is what I call it. And as far as I know, it's the only kinase that has this tryptophan here in the active site. And so that sort of brought home that it's very messed up pseudokinase. But once we purified this, we wanted to do some activities to try to understand it. And although there have been some reports in the literature, we sort of first confirmed in our own hands that it does not in fact have any activity. So we knew that there are a number of pseudokinases that, although predicted not to have activity, actually were found to be active kinases and sometimes had evolved sort of alternative mechanisms to develop activity. In this case, just to give you an example, we compared it to ULK1, and so even for autophosphorylation, you can see it just has basically negligible activity. For autophosphorylation, which in general is sort of a very low bar for activity, you can sort of force any pseudokinase to autophosphorylate if you give it enough radiolabeled p32 ADP. But the big question is whether it can bind ATP, and there were some reports that it could, in fact, bind ATP. So the question is, how can you study a dead enzyme? And first, just to look for binding, you need to develop assays for nucleotide binding, sort of the best approach to see if it can bind ATP, and then to use that assay or other assays to then study its activity and find inhibitors. And so throughout the literature, there are a number of different assays that people have developed. And so I'll go through a couple of them that we've sort of adopted for ULK4. So one of the ones that I came across is using a fluorescently labeled ADP or ATP that has a very small fluorophore called MANT or TNP. And these are sort of environmental fluorophores in that they're so small that actually their fluorescent properties change based on the local environment of the molecule. And so depending on whether this ADP is bound to the protein or not, sometimes you can see changes in fluorescence. So we tried both of these for ULK4, but we did not see any changes based on the local environment. However, I also found in the literature that the MANT fluorophore can also be adapted for a FRET assay. So instead of looking at the MANT excitation and emission, you can actually use the 280 excitation of the protein and then detect a FRET signal based on the emission from the MANT ADP. And so this is based on the 280 excitation, which, as you know, mostly comes from tryptophans. And we have that tryptophan right in the active site of ULK4. And indeed, we were able to see a nice FRET signal using this MANT nucleotide. And we also confirmed some earlier reports that it does, in fact, bind without magnesium. So it binds actually better without magnesium. And then using this assay, we're also able to use this as a probe to then use normal ADP and ATP. And we found it actually binds quite tightly. So it binds sub-micromolar to ATP without magnesium. So it's actually one of the tightest binding pseudokinases for ATP, which begs the question of, why has it maintained this very strong binding activity for ATP, even though it seems to have sort of messed up every important aspect, at least for catalyzing the transfer of phosphate. And then the other assay we used that I think we talked about earlier today was this ADP biotin. ATP biotin is another common probe for kinases and pseudokinases. And so basically it relies on the fact that you have a lysine in the active site, although we're missing the sort of canonical lysine, that it will, if it can bind ADP or ATP, it can attack this probe and then form a covalent amide bond with your desthiobiotine. And then once you have that covalently labeled, then you can detect it through a number of ways. In this case, we used basically a Western blot, so using a streptavidin, a fluorescent streptavidin to look for binding. And we can use this to look at first different mutants of the protein. So based on our structure, we wanted to try to understand how it binds ATP. And so we mutated a number of amino acids, including some control experiments. So if you don't have this ADP biotin, you don't see the signal. If you add EDTA, you don't really see loss of the signal like you would with a normal kinase, confirming its dependence on, or its independence of magnesium. And so a few amino acids that really struck out. So this 139 residue, that is the one that replaces the DFG, the D in the DFG. And so that seems to be essential for binding nucleotide. And we also looked at a number of basic amino acids. So because it's metal ion independent, typically other kinases have evolved like a lysine or arginine to sort of replace that magnesium in order to counteract the negative charge of the diatriphosphate. And so we looked at a couple of them. And in this case, with this ADP biotin, we found that only the lysine 126 was required for binding the nucleotide. And after our structure, Stefan Knopp's group also solved a nice structure of ULK4, in this case with ATP gamma S. So it really helped shed light on the ATP binding mechanism. So they could see ATP binding in the active sites, and they sort of also saw that it has this very unusual binding mode, including this leucine that I mentioned that sort of helps pack up against the active sites. And at least for their ATP, they can see arginine playing an important role in binding to that, to the gamma phosphate or thiophosphate. And they also did some very nice evolutionary analysis on ULK4. And a cool thing that they found was that in addition to the conservation of this binding site, is that they found that there were actually other alterations that sort of co-evolved with the degradation of the active site, including this extended insertion into the activation loop. So if you look at the ULK family, ULK4 alone has this sort of large insertion. And they hypothesized that that might help sort of stabilize the alpha C helix to help sort of protect that ATP binding site. And so I'm going to tell you about sort of the major area we're interested in ULK4 is to develop small molecules. So sort of a chemical biology approach for understanding the role of ULK4. And so we already developed this assay to look at binding a nucleotide. And so we can use this assay to look for compounds that can compete with these labeled nucleotides for binding. And so in order to get at compounds, we took a number of approaches. So the first was to do a virtual screen, which is sort of the easiest from my point of view because it doesn't cost us anything. And so my next door neighbor here is Avner Schlesinger, who is a computational chemist, and he specializes in kinases among other proteins. And so he helped us do a virtual screen using the Zinc database and went through millions of compounds and identified some that sort of based on our pharmacophore for our ULK inhibitor that sort of had some of the key motifs. And then we cherry picked some of the top docked compounds and then use our MANT assay to measure binding constants. And through this, we were able to identify a couple of sort of double-digit micromolar compounds that can compete with the fluorescent nucleotide binding, which is sort of a decent starting point for kinases, given that these are sort of lead-like scaffolds and not sort of full compounds. And the other approach we took was to do sort of a mini high-throughput screen using known kinase inhibitors. So using a collection of commercial kinase inhibitors, we use the same fluorescent screen. And in this case, these are sort of more developed compounds. We were able to get single-digit micromolar compounds that bind to ULK4. And we used our ADP biotin assay as an orthogonal assay to confirm that we are in fact inhibiting nucleotide binding and that those compounds were not just interfering with the fluorescent assay, since that was our first readout. And you can see that, especially with this two-color assay, it's easy to see which compounds are competing best with the nucleotide. And indeed, these compounds here, this R788 prodrug and PHA compound, I'll show you a little more data on the CDK2 inhibitor, are sort of our best compounds. And so subsequently, we were able to crystallize ULK4 with this PHA, this Pfizer compound that's a CDK2 inhibitor, and we were able to see nice density for it. And we can see some of the contacts, which are actually not too many in the active site, including the hinge binding and also this contact to the acetamide here. So I mentioned this PHA compound is a CDK2 inhibitor. And so we looked at the structure of CDK2 bound to this compound, which is publicly available. And you can see it binds in a very similar conformation, especially in the sort of the proximal site here. And this sort of highlights one of the main challenges with screening from kinase libraries and with pseudokinases is that these are compounds that are already sort of optimized towards other kinases. And so we're going to have to remove, for example, in this case, CDK2 inhibition and then make it more potent towards ULK4. So with any of these initial hits, we're going to have to do medicinal chemistry to try to optimize it towards our target pseudokinase and away from other proteins. But on the plus side, we have an assay and we have a high resolution structure. And it's actually pretty easy to purify and crystallize. And so it sort of is amenable for doing some structure-based drug design to sort of get to a compound that's selective enough to use in cells. Because right now, none of our compounds are really selective enough that we can confidently say that the effect that we see in whatever phenotype is due to its ULK4 binding. So then let me just tell you in the last few minutes about another strategy that we've developed more recently for finding inhibitors of pseudokinases for ULK4. So one of the main drawbacks of this MANT assay is that we're looking at excitation in the 280 nanomolar range, which unfortunately is where a lot of small molecules autofluoresce. So it's pretty common, basically anything that has something that looks like a tryptophan. So actually including our ULK1 inhibitor that was in our crystal structure. So we were never able to measure the binding constant of the inhibitor that we crystallized the protein with, because it fluoresces in that same region, and so we just see a very high signal. Another drawback with a lot of these assays in general, which is sort of a problem with all pseudokinases, is that, or pseudokinase assays, that it often requires a very high concentration of protein because it's sort of a stoichiometric assay. So rather than getting amplification from your enzyme, your assay is usually dependent on, the signal is over the noise, it's dependent on the concentration of your enzyme when it's bound to nucleotide. So it uses up a fair amount of protein and sort of problematic sometimes for finding inhibitors if your enzyme concentration is very high. So this other assay we developed, a fluorescent polarization assay, which a lot of you will be familiar with. It's a very common assay used kinases and many other enzymatic assays, but basically relies on a fluorescent compound having different polarization properties depending on whether it's free in solution or bound to a protein. So if it's free in solution, the compound can rotate, can tumble easily, and so you get a low polarization signal. And once it's bound to your protein, because the protein is a much larger molecular weight, the complex tumbles more slowly, and then you see increased polarization. And then if you have an inhibitor bound that displaces the fluorescent probe, the probe is now free in solution, and then your polarization signal decreases again. And so we use a fluorescent ATP molecule. In this case, we use fluorescent BODIPI ATP. And using this, we were able to titrate the ULK4 protein and get a nice binding curve. And we measured a binding constant of about 1 micromolar, which is consistent with our other assay too. So it shows that indeed this assay works. And so the big advantage here is that this, for the fluorescent polarization, you can use any fluorescent wavelength. So this BODIPI, which is basically a fluorescein type fluorophore, is much further away from the autofluorescence of most compounds. And so we didn't get any false sort of false negatives in our assay based on this wavelength. And so we redid our screen, the same commercial compound of kinase inhibitors, and we identified several new inhibitors that were previously blocked based on our MANT assay. And so we're now working on these new compounds to try to measure how potent they are. However, this still requires somewhat high concentration of protein, but it sort of overcomes that first barrier. And so just to summarize where we are, the main goal is to improve inhibitor selectivity. So we've sort of overcome all the assay development limitations, and now it's a medicinal chemistry problem in order to get selectivity, which is sort of the main challenge with kinases and pseudokinases, especially if you're starting off with sort of a weak binder to the active site. And I didn't talk about too much, but sort of overall goal is to really try to understand its role because we don't know too much about it. So the inhibitor approach will take some time to get selectivity. So another approach is to use genetics to try to understand the role of the ATP binding pocket, which we know it must be important because it's conserved and binds so tightly. And another aspect that a lot of the speakers have mentioned is this idea that pseudokinases really exert their effects by partnering with other proteins. Oftentimes it's an active kinase, and so the ATP binding of the pseudokinase can help regulate the activity of another kinase. And so Stefan's group, and then we have also been interested in trying to identify possible interaction partners for the protein and potentially in disease contexts. And so this sort of future goals for trying to understand this kinase, because it's really sort of the early stages of this protein. We don't really know its biological function. So with that, just want to thank the lab. I want to highlight Shishmita here as a postdoc, and she did most of the experiments for ULK4. Here's a picture of our early stages of our group. This is during the eclipse from a few years ago that came across New York on our rooftop. And I want to thank my collaborator, Avner, and my funding sources. And I want to thank you for your time. And thank you for inviting me here, and I'd be happy to answer any questions.
Thank you, Michael. It was great. We have a couple of questions, but let me get started. So Are you using the sort of the variations in the active site of ULK4 in your design of these compounds? Like you mentioned the tryptophan that is sort of unique to the ULK family. So are you using that in any way in your design?
Yeah,
compound?
that's a good question. And so that's sort of the idea. So we haven't done any of the medicinal chemistry yet, but that will sort of be the basis, try to take advantage of some of the weird effects of the protein that will help us. It's actually, yeah, kind of interesting that some of the same compounds bind to our kinase as to other kinases, even though everything is so messed up. And so we have some ideas based on some of the other compounds that we can take advantage. The other major thought is because it has this lysine and arginine, instead of having the magnesium aspartate, then maybe we can find a compound that sort of has a negative charge on it that can interact with the positively charged amino acids that maybe will interfere with just like the aspartate and other normal kinases.
Right.
But that's definitely one of the goals. If we can do it rationally to try to take advantage of this weirdness.
There's a question from Francisca on the conformational differences between the ATP bound form and the compound bound form of the ULK4. Do you see any obvious differences that you want to comment on?
That's a good question, and you can allow Stefan to chime in too if he's interested. But as far as I could tell, it looked pretty identical. There was there was different crystal form between them, but overall they were both sort of in that active conformation. And so there were some, I guess, some ideas that maybe we could find a compound that maybe was like a type 2 inhibitor. That's sort of a overarching goal, I guess, in the maybe in the pseudokinase field, to come up with compounds that can induce an inactive pseudokinase conformation versus an active one. And so maybe we could induce an inactive conformation with some compounds. But so far, it looks like all the compounds are sort of in that same ATP binding conformation.
Okay. Thank you, Michael. I think we're sort of nearing the hour, so I think we should probably just try and conclude this webinar. I think it's been fantastic with all those speakers sort of highlighting various aspects of pseudokinase biology and beginning with Pat really highlighting the challenges in studying the darkest of the pseudokinases, PSKH2, and James Murphy nicely highlighting how one can use nanobodies to really probe pseudokinase and dark kinase functions, with Ben highlighting how he's using his proximity networks as a way to probe putative sort of scaffolding functions of kinases and pseudokinases. And finally, Michael nicely demonstrating how structural biology and medicinal chemistry and the unique variations in the active site can be used in developing selective inhibitors. So I think we really highlighted with this webinar the new challenges and opportunities sort of moving forward. And again, I want to thank all the speakers for taking the time out of their busy schedule to really highlight this. So I want to kind of hand it over to Stefan to say some sort of final thank you notes. And I think particularly the Target 2035 staff who really done a phenomenal job of coordinating all of this. Stefan, do you want to conclude?
Yeah, Kanan, thanks a lot. You summarized it really nicely. There is not really much to add from my side. I mean, I only want to mention once more the Target 2035 initiative. So please try to stay engaged. And I think it would be really nice if we can resolve the problem of generating high quality inhibitors for the druggable proteome as a global initiative, and see that we kind of combine our efforts and get that tackled. And I mean, I always see, I mean, by sampling these libraries that most compounds are not available to the public. And I think this is a big problem, because if they're really interesting, they're synthesized somewhere, and sometimes without good quality control. So as a chemist, I think it should be one of your foremost concerns that actually compounds are of good quality and what you actually published. So if at all, I think that would be a good reason to, and I hope that in the future we would have more pseudokinase inhibitors coming up. Certainly really interesting area. So I just want to thank all speakers again, and in particular James, who must be really tired now, but he still looks pretty fresh. So I think you do this more often. Living in Australia, so I think it's one of the common problems. So thanks also everybody for staying until the end of the meetings. I saw we had close to 120 participants. Now it shrunk to 60, but I think this is pretty good for considering it's quite late for some people. Yeah. Okay. So I hope you see you all one of the upcoming meetings.